Hey everyone, in this video we are continuing with all the material we have talked about for nucleic acids, only this time we're going to focus on the lens of Unit 2 and how what we talked about in Unit 2 is still relevant to helping us understand what is going on in Unit 5. So in this video we are going to compare and contrast prokaryotic to eukaryotic DNA, and I'm going to do that using a T-chart. And it's important when we do this comparison that we always talk about both sides. This is something that students frequently miss on FRQs. They'll tell me the characteristics of prokaryotic DNA, but then they forget to contrast that to how the eukaryote is different. So this line right here is to help you represent that this would be kind of like a however, or what we call the but. So if we're talking about the shape of prokaryotic DNA, then this line is to help us remember that we also need to include the shape of eukaryotic DNA. So this space is going to contrast directly with this space, and it's important that you keep both of those, or you mention both of those on any free response questions. So the first thing we're going to talk about is the basic structure of prokaryotic and eukaryotic DNA, but it's important that you all understand that in both cases, prokaryotes and eukaryotes, we're still talking about DNA. Many students want to say that prokaryotes have RNA, and that's true, but that's not their genetic information. In both eukaryotic and prokaryotic cells, both of them store their genetic information as DNA. So that's something they have in common. Both of them store their genetic information as DNA, but the structure of that DNA and the location of that DNA is what's different. So let's start with the location of the DNA, because that's one of the biggest defining features of a prokaryote and eukaryote. In prokaryotes, it's located in a nucleoid, which does not have a membrane around it. It's just a general area. Whereas eukaryotes do have a double membraned envelope. Another way that we can contrast prokaryotes to eukaryotes is the shape of the DNA. In prokaryotes, it's going to be circular, while in eukaryotes, it will be linear. Another way to contrast the prokaryotic to eukaryotic DNA is by the amount of DNA. Typically with prokaryotes, they're going to have one single chromosome. And even if they did have more than one, they're identical chromosomes. They just have multiple copies of the same thing. Whereas in eukaryotes, they will have usually multiple chromosomes. We can also add to this that prokaryotes will contain less genetic information compared to eukaryotes. And because of the different amounts of DNA between prokaryotes and eukaryotes, the way that they have to be organized is also going to be different. So prokaryotes are going to not contain histones. Histones are proteins that bind to the DNA and help to keep it from getting tangled up, and also aid in whether or not the DNA is going to be expressed into proteins. Eukaryotes will have a tremendous amount of histones. And these histones help to create the structures that we talked about with DNA when we talked about chromatin versus chromatid. It's those histones that are going to aid in that process of creating the supercoiled chromatids. Now, prokaryotes do have some proteins that help with coiling, but they are not histones. They're just kind of an archaic form of a protein, and it doesn't coil the DNA to the extent that you see in eukaryotic cells. A couple of other topics that are worth mentioning is the idea that in prokaryotes, they are able to have external DNA incorporated into their own, or pieces of their own DNA that are broken off and floating around in the cytoplasm, and these are called plasmids. Eukaryotes do not have plasmids. They don't have external DNA that they use beyond what's within the nucleus or the mitochondria or chloroplast, but they what makes eukaryotes different is that they do have introns. And introns are non-coding DNA that is within our genetic code. And they're just large segments of DNA that don't actually make a protein. And that's something you don't see in prokaryotes. Prokaryotes have no introns, but it's worth mentioning that prokaryotes do have plasmids, which are external pieces of DNA. I hope this video was helpful. Thanks for watching. Bye.